Hello. Okay, the student who have joined, please can you write in the chat box if my voice is clear and my presentation is visible. So we will wait for 1 or 2 minutes and then we will start today's session. Okay, welcome to you all to this week 10 live session of the NPTEL course Organic Chemistry in Biology and Drug Development. I am Moushumi Deb and I am the PMRF Fellow of the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So this is a week 10 live problem solving session in which we will discuss some 10 to 15 questions related to your week 10 course content, and this type of question will help you to understand the course better, and also it will help you to do this week's assignment and for your finals exams also. So we will start today's session. Okay, somebody wants to join. Okay, so we will now start today's session, and during the session if you have any doubt, anything, please write in the chat box. I will answer those queries at the end of the session, and still if you have some doubt after the ending of the session, you can also write a mail to me. I will share my email ID at the end of the session. Okay, so let's start today's session. If somebody joins later, we will just allow him or her to join the meeting. So I think my voice and my presentation both are clear, and if you cannot hear me or you have difficulty to view the screen, please write in the chat box. Okay. So the first question, the problem number 1, it says that which of the following is first known antibacterial agent, and the options are option A penicillin, option B urea stibamine, option C salvarsan, and option D morpholin. So you have to identify the first known antibacterial agent among these 4. Okay. Okay, someone is saying that the correct answer is A. So let's see which is the correct answer. Okay, so actually the correct answer for this question is number C salvarsan. It was the first discovered and first known antibacterial agent. Okay, this salvarsan is basically used for the disease syphilis, and I will show you the structure of the salvarsan. So this is the structure of the salvarsan. Here two arsenic are joined by a double bond, and with this there is a orthoaminophenol kind of moiety. And this salvarsan, it is a very unique molecule because we all know that arsenic is harmful for human body, but it itself is a drug molecule, although having the arsenic as a core center. And this is the existing form of the salvarsan. This has a pentameric structure where the arsenics are joined with each other, and we have this pentameric structure of the drug salvarsan. So this is the first known antibacterial agent. Okay, so I think it is clear. I will move toward the next problem. Okay, the problem number 2, it says that which of the following is not a semi-synthetic antibacterial agent, and the options are as follows. Number A ampicillin, option B is carbenicillin, option C cephalexin, and option D sulfonamide. So you have to identify among these 4 which is not a semi-synthetic antibacterial agent. So let's see first which is the answer, and then we will discuss what is a semi-synthetic antibacterial agent. Okay, so the correct answer is sulfonamide. It is not a semi-synthetic antibacterial agent. Now let's see the structure of sulfonamide. So this is a structure of sulfonamide. So we all know what is amide. This the amide is the, like, amide has a structure like this. See, there is a C, here is a double bond O, and here is NHR or NR2 or NH2. So if we have a carbonyl group and nitrogen with together, it is called amide. So what is a sulfonamide? Sulfonamide is nothing but if we have a sulfur in place of carbon. You can see there is a sulfur group. There is a sulfur with two oxygen molecules, and in place of carbon. So it is called the sulfonamide, and it is a synthetic antibacterial agent. It is not a semi-synthetic antibacterial agent. Now, what is the semi-synthetic and synthetic and natural? So antibacterial agents, they can be of three types. So first one is the natural antibacterial agents. So they are we get this natural antibiotics mainly from fungal sources. Okay. Now the original antibiotics were derived from fungal sources. They can be referred as a natural antibiotics. Maybe these can be produced also in our laboratory, but initially they were mainly isolated from the natural fungal sources. So they are called natural antibacterial agent, and the examples are benzyl penicillin and gentamicin. These are so few of the natural antibacterial agent. Now what is a semi-synthetic antibacterial agent? They are chemically altered natural compound developed to decrease the toxicity and increase the effectiveness of the natural compound. The examples are ampicillin and amikacin. Okay. So they are the semi-synthetic. They are called the semi-synthetic, which are, they are chemically altered natural compound. Means there is a natural compound, antibacterial compound present, but due to the toxicity and less effectiveness, they need to be chemically modified in laboratory, and they are then called the semi-synthetic antibacterial agent. And ampicillin and amikacin is the examples of semi-synthetic antibacterial agent. Now what is a synthetic? The synthetic antibacterial agents are chemically designed in the laboratory. Bacteria are not exposed to the compounds until they are released. They are also designed to have an even greater effectiveness and less toxicity. And moxifloxacin and norfloxacin are synthetic antibiotics. So is sulfonamide. Okay. So please remember the types, the classification of this antibacterial agents. There are three type of antibacterial agents. There are natural antibacterial, semi-synthetic and synthetic, and this is the sulfonamide we have, which is a synthetic antibacterial agent. And please, I will suggest you, please remember 2 to 3 examples of each class of the antibacterial agents. Okay. So I think it is clear what is a synthetic, what is a semi-synthetic antibacterial agent. We will then move toward our next problem. So problem number 3, it says that which of the following statements is not true for bacteriostatic agents. Okay, the options are here. The option A, it says that the agent stop the growth of the bacteria. Option B, the bacterial population remains same. Option C, the agents destroy the bacteria. And option D, the agents inhibit the processes like replication, translation and transcription. So you have to identify the false statement, which the question has asked of which is not true, means the false statement regarding the bacteriostatic agents among these four. So let's see the correct answer, then we will discuss what is the bacteriostatic agents. So the correct answer means the false statement is the agents destroy the bacteria. So what is the bacteriostatic agents? These antibiotics that stops or inhibit the growth of bacteria. That means no population generation of bacteria grows, but they do not kill the bacteria. They show their growth and slow their growth and reproduction. They inhibit the DNA replication and protein synthesis of bacteria, work with the immune system of the host. Minimum drug concentrated is required. And for example, tetracycline, penicillin, sulfonamides, linazoloid, chloramphenicol, clindamycin and macrolides. These are few example of the bacteriostatics agent. So there is another antibacterial agent which is called bactericidal. So there are two actually. The one is bactericidal and one is bacteriostatic. So what is bactericidal? The bactericidal refers to antibiotics that kill the bacteria. But bacteriostatic refers to the antibiotics that prevent the growth of the bacteria. In the first case, the action is irreversible, but for the bacteriostatic, the action is reversible. The bactericidal, they inhibit the cell wall formation of the bacteria, but the bacteriostatic, they inhibit the DNA replication and protein synthesis of the bacteria. Bactericidals do not work with the immune system of the host. Bacteriostatics work with the immune system of the host to prevent the growth and the reproduction of bacteria. The MBC refer to the concentration of the drug required to kill 99.9% of bacterial population in case of bactericidal ones. The MIC is the minimum drug concentration or minimum inhibitory concentration which inhibits the bacterial growth, and the MIC value is it is related with the bacteriostatic antibiotics. Okay, and the examples of the bactericidal is the beta-lactam antibiotics, cephalosporins and vancomycin. And the examples of the bacteriostatic is the tetracyclines, splinomycin, chloramphenicol and sulfonamides. Okay. So, this is a very important comparison of the bacteriostatic and bactericidal agents. So, please remember each and every point is important in this case, I will suggest, but remember that bactericidal, they basically kills the bacteria, and the bacteriostatics are the one who do not kill the bacteria, but they inhibit and slow down the growth of the bacteria. Okay, so this is a very important point and very important question. Please remember and this carefully. So, I hope you have understand this question. The first options, the first two options are correct. The number C is incorrect, and the number D is also correct. Okay. So, now I will move toward the next problem. If you have any doubt, any problem, please write in the chat box. I will try to answer those at the end of the today's session. Okay. And if you cannot hear me or see the presentation, please write in the chat box, and this presentation and all this recording, it will be shared to you in due time by the NPTEL side. So, problem number 4, it says that penicillins exert their antibacterial action by inhibiting cell wall biosynthesis. Which cell wall biosynthetic enzyme is inhibited? Number A, carboxypeptidase. Number B, transpeptidase. Number C, glycosylase, and number D, phosphatase. So, we all know penicillins are a very important antibacterial agent, and they actually exert their antibacterial action by inhibiting the cell wall biosynthesis. The bacterial cell wall biosynthesis, it is basically hindered by the penicillins. Okay. Now, there is an enzyme which is actually helps bacteria for the cell wall biosynthesis, and penicillins exactly inhibit that biosynthetic enzyme. So, you have to identify which one is the correct biosynthetic cell wall biosynthetic enzymes which is inhibited by penicillin. Okay, so let's see the correct answer for this problem. So, the correct answer is number B, transpeptidase. So, this transpeptidase enzyme, it is present in the bacteria, and which helps its the synthesis of the bacterial cell. So, you can see there is a beta-lactam mechanism of enzyme. The beta-lactam, it is actually the active site of the penicillin antibiotics. If you know the structure, what is the beta-lactam? So, actually lactams are suggest the cyclic amides. So, the cyclic amide are known as the lactam, and for in case of penicillin, there is a beta-lactam. So, the beta-lactam means there is this kind of structure, active site structure. So, this is the structure of the beta-lactam. Why it is called beta? Because it is a 4-member ring, and there is a alpha carbon here and a beta carbon here. Okay. So, there are 2 carbons actually. So, this is called the beta-lactam ring, and this is the active site, and it is necessary for the action of the penicillins. So, there is a transpeptidase, as you can see here, there is a transpeptidase subunit present, and this block of the transpeptidase activity interrupts the cross-linking and the cell wall biosynthesis in the bacteria. So, there is a as you can see, there is a terminal d-alar residue of this case, and it is inhibited. This thing is broken by the beta-lactams because they bind to the transpeptidase active site. So, like every enzyme, the transpeptidase also have a very good active site which is necessary for the action and the activity of the enzyme. but beta lactams, they bind to the active site of this transpeptidase, and thus they inhibit the cell wall biosynthesis and the cross linking of the cell wall for the bacteria. Okay, please remember this is a very important enzyme, this transpeptidase, and this is a very important topic because beta lactams are a very important class of antibacterial agents. So I will move toward our next problem. So the problem number 5, it says that the three simple building blocks of penicillin are option A glycine, cysteine and valine, option B alpha amino adipic acid, cysteine and leucine, option C beta amino adipic acid, cysteine and isoleucine, and option D alpha amino adipic acid, cysteine and valine. So the correct answer is for this question, the option is option D, alpha amino adipic acid, cysteine and valine. These three are the simple building blocks of penicillin. So this is the penicillin molecule. As in the previous question, I was discussing that there is a beta lactam ring, which is called the beta lactam ring. Lactam means the cyclic amide, and it is a amide because nitrogen is connected with the carbonyl moiety, and this is the beta lactam group, which is very important for this penicillin antibiotic, and it is called the beta lactamase class of antibacterial agents. Okay, because there is a beta lactam group is present. So, and please remember the structure of penicillin. So I generally say that you don't need to remember the big structure and big molecule, but penicillin is a different thing. Penicillin is a very important molecule, and I will suggest please remember the structure of penicillin, including this stereospecific areas. Means there is a hydrogen below the plane here, there is a carboxylic acid below the plane here, and a amide group above the plane. So this stereospecificities is very important for the activity of the penicillin, and these are the three very simple building blocks of penicillin. There is a L amino adipic acid, and there is L cysteine and D valine. So by this way the penicillin biosynthesis is happens, and there is a first there is a ACV tripeptide formation, and after that there is isopenicillin N formation, and from this there can be cephalosporins can be formed also, and we have a penicillin G. So there is a two type of penicillin, one is penicillin G and another is penicillin V. So if it has this benzylic here, if R stands for CH2 pH, then it is called the penicillin G. Okay. So it is very important, though you don't need to remember this each and every step, plus please remember the structure of the starting materials, because it is very easy, because L-cysteine is an amino acid, we all know it is a alpha amino acid which has a CH2 ACH group, and D-valine is also alpha amino acid which has a this kind of allyl group here, and there is a L amino adipic acid present. This is a adipic acid which is a dicarboxylic acid, and in one alpha position there is a amino group, so it is called a alpha amino adipic acid. Okay, so I think it is clear what is the three simple building blocks of penicillin, and this is we will discuss it later again when we will discuss the isopenicillin N enzymes, then we will discuss it again. Okay, so I will quickly move toward our next problem. So the problem number 6, it says that which of the following statements is true regarding the gram positive bacteria. So there are two kind of bacteria, as we all know. Mainly there is gram positive and gram negative bacteria, and you have to identify the true statement regarding the gram positive bacteria, and the options are as follows. Option A, they are very much susceptible to antibiotics. Option B, they have low lipid content. Option C, peptidoglycan layer is thin and majorly single layer, and option D, outer membrane is not present. Okay, so you have to identify the true statement regarding the gram positive bacteria here, and the let's see the correct answer first, then we will discuss the gram positive and gram negative bacteria. So the correct answer is the peptidoglycan layer is thin and majorly single layered. So this is the incorrect this is the true statement, this is the true statement regarding the gram positive bacteria. Now let's discuss the gram positive and gram negative bacteria and their differences. So there are several parameters based on which we can differentiate the bacteria into gram positive and gram negative and we can say which is gram positive and which is gram negative. And the first parameter is the cell wall. So if the cell wall is single layered, smooth, and single layered and smooth, so then it is the gram positive bacteria. Okay, and if it is the double layered and wavy cell wall, then it is the gram negative bacteria. So it is our correct answer, because the peptidoglycan layer is thin and majorly single layered, which is the correct statement. So we can see it in the first first point of this table, that the for gram positive bacteria the layer cell wall will be single layered and smooth. So cell wall thickness, the thickness of the cell wall is 20 to 80 nanometers in case of gram positive bacteria, wherein in the in the gram negative the cell wall is 8 to 10 nanometers, the thickness of the cell wall. Okay, the peptidoglycan layer, it is a thick layer and also can be multi-layer, and it can be also thin layer and often the single layer in case of the gram negative bacteria. And there is a teichoic acid, the teichoic acid are present, and the teichoic acids are not present in gram negative, and lipopolysaccharide is not present in gram positive, the lipopolysaccharide is present in gram negative, and the outer membrane is not present for the gram positive, but the outer membrane is mostly present in the gram negative. The lipid content is very low, and the lipid content is the lipid content is 20 to 30%, and they are very susceptible to antibiotics and they are resistant to antibiotics. Okay, I think okay, I think there is a typographical error in this question, because there is a there it should be actually false regarding. Okay, I am so sorry, I will just correct it. The statement, it will be false. I will just correct it here. Okay, so there is a error in this question, I am sorry about it. So the correct, actually the first this for the gram positive bacteria, the question should be the what is the false regarding the gram positive bacteria, because in case of gram positive bacteria, the peptidoglycan layer, it is thick and can be multi-layered, not thin and single layered. It is for the gram negative bacteria, and other 3 points are correct. So the question was incorrect, I have just corrected it. So the question will be, which is the false regarding. Okay, so the question, it will be, which is the false regarding the gram positive bacteria. Okay, and the first point is correct, because they are very susceptible to antibiotics, and the number B is also correct, they have low lipid content. Number D is also correct, the outer membrane is not present. The peptidoglycan layer is thin and majorly single layer, it is the false statement. Okay, because as you can see in the third point, for the gram positive bacteria, the peptidoglycan layer, it is thick and can be multi-layered. Okay, so there is a there was a mistake, I have just corrected it. So this is a very important point again, because the gram positive and gram negative bacteria are very important, and you have to remember the differences in their parameters. Okay, because yeah, it can be sometimes, like I have made a mistake here regarding this question. You can also make a mistake, so please remember the point, so that you do not make any mistakes in your exams. Okay, so I think it is understandable now, there is no problem regarding this question, and we will now move toward our next problem. So problem number 7, it says that which reaction is catalyzed by beta lactamase enzyme, and the options are: number A, the final cross-linking reaction to form bacterial cell wall; the hydrolysis, option B, the hydrolysis of acyl side chain of the penicillin; option C, the hydrolysis of 4-membered ring in penicillin; and option D, the biosynthesis of penicillin from LLD ACV. So which of the following reactions is catalyzed by beta lactamase enzyme, you have to identify, and the correct answer is the hydrolysis of 4-member ring in penicillin. So there is this beta lactamase enzyme. So I have shown you the structure of penicillin in the previous slide. So this is the structure of penicillin, and I told you for the activity, this beta lactam ring is very important. So but this beta lactam ring, it is hydrolyzes by get hydrolyzed by this beta lactamase enzyme, and after hydrolysis we have this carboxylic acid and amine here. Okay. So this is the mechanism how the this 4-member ring get hydrolyzes. First we have a base here. This base deprotonates a nearby serine residue, and after the deprotonation we have a O minus on this serine side hydroxyl group, and this O minus will attack this carbonyl carbon here, and we will have O negative due to this resonance, and we have a serine attached here, and after that what will happen, this negative charge gets back here, and this bond, this carbon nitrogen bond got broken here. Okay. Then we will have a water molecule here which attacks to this carbonyl carbon, and after the attack of this water, and which is actually deprotonated by a second base here, and this after the deprotonation we will have a OH minus which will attack this carbonyl carbon again, and just like that now the serine moiety, it will get released, and we will have a carboxylic group here with a amine group here. And okay, so this is the overall mechanism how this 4-member ring get hydrolyzed, and the beta lactamase enzyme this catalyzes this hydrolysis of the penicillin rings, this 4-member ring of penicillin. Okay. So I think it is clear. Please remember this mechanism here, because this mechanism is very easy mechanism. This because as you can see there is nothing, there is some just attack on this carbonyl group at first, the breakage of the carbon nitrogen bond, and then this attack happens by the serine molecule, and after just by the attack of the nearby water residue on this serine attached carbonyl group, and the serine got we get back this serine molecule at the end of the catalysis cycle. So please remember this catalytic cycle, it is very important, and also it is very easy to remember this how this 4-member ring gets hydrolysis. Okay. So I think the mechanism is clear. I will move toward the problem number 8. Okay, so the problem number 8, it says that which of the following statements is false regarding the NDM bacteria. So the option A is NDM stand for New Delhi metallo beta lactamase enzyme. Option B, they are class B lactamase enzyme. Option C, they hydrolyzes oxacillin. And option D, it utilizes a metal ion to hydrolyze beta lactam ring. So you have to identify the false statement among these 4 options regarding the NDM bacteria. So let's see the first correct answer, then we will discuss what is the NDM bacteria. So the correct answer is they hydrolyzes oxacillin. So it is the false statement regarding this NDM bacteria. So what is a NDM bacteria? And so it is a New Delhi metallo beta lactamase 1. The full form of NDM is New Delhi metallo beta lactamase. Okay, so NDM New Delhi metallo beta lactamase 1 or NDM 1 is a newly described metallo beta lactamase MBL, which is metallo beta lactamase, and first identified in 2008 in single isolates of K. pneumoniae and E. coli, both recovered from a patient repatriated to Sweden after treatment in hospital in New Delhi, India. Like other acquired MBLs, NDM1 hydrolyzes all beta-lactam antibiotics except azithromycin, which is usually inactivated by co-produced extended spectrum or MPCB beta-lactamases. Okay. So this is all about the new Delhi metallo beta lactamase, and let's discuss some new things about here. We will discuss the classification of the beta lactamase enzymes. So this is called Ambler's classification, and it is based on the active site. So if the active site has a metal site like zinc 2 plus or metallo beta lactamase, then it goes to the class B. Okay. And if there is a serine non-metal side or serine beta lactamases, they have 3 classes. One is class A, there is class D and class C. So what is there is this metallo beta lactamases, that is the NDM bacteria, NDM lactamase, they follows this class B, and they are called group 3. And there is a class C which is AMPC beta lactamases, and the class D they are oxa beta lactamases, and the class A they are ESBL. Okay. And there is also a classification which is BJM classification, where this class A and class D are group are combinedly called group 2, and the class C called group 1, and the class B called group 3. So you don't need to remember this group 1, group 2, group 3, but remember the class B, class C and class D. And in the class B there is a metal side zinc 2 plus metal, because of that we call it metallobetal actamase. And in the class A, C and D there is a serine active site and there is no metal ion, and this is called serine beta lactamases. Okay. So I think the classification is clear, and the what is the NDM, it is also clear here. So again I am saying, if you have any doubt regarding any problem, please write in the chat box. At the end of the session I will try to answer your queries. Now problem number 9, it says that augmentin is a life saving drug, is a mixture of option A oxacillin and ampicillin, option B amoxicillin and clavulanic acid, option C oxacillin and clavulanic acid, and option D the amoxicillin and pyruvic acid. So you have to identify the what are the correct combination for the drug augmentin. It is an antibacterial drug, and let us see the answer first, then we will discuss this augmentin. So the correct answer is amoxicillin and clavulanic acid, which is the option number B. So these are the structures of the amoxicillin and clavulanic acid. So the amoxicillin, it is also a beta lactamase, there is a beta lactam enzyme present. So and there is a side chain of this, I have shown you in the case of penicillin, there is a in after there is a CH2PH, and here we have alpha, there is a amine, and there is a phenol type of moiety here, and this is the structure of amoxicillin, and this this part is the same is as the penicillin, and this is the clavulanic acid. We also have a beta lactam kind of moiety here. Okay. And this augmentin is a life saving drug, which is mainly, actually it is a also a antibacterial drug, and you can see there is mentioned that it is amoxicillin and potassium clavonate tablets. Potassium clavonate, when there is a potassium, actually it does not there is the clavulanic acid, it actually in the mixture it says as a salt form, is a potassium salt form, and it is a very important mixture. You don't want to remember the structures here, just remember the names, like the amoxicillin and clavulanic acid, which are the main components of the life saving drug augmentin. Okay. So I will now move toward our next problem. Okay. The problem number 10, it says that identify the correct statement among the following. Option A, PLP dependent isomerism takes place when D amino acid is converted to L. Option B, alanine racemase is inhibited by D-cycloalanine. Option C, D-cycloserine is used in first line of treatment of tuberculosis. There is a typographical here error here. I will just okay. I will just okay, it should be tuberculosis. I will correct it here. Okay. Okay, pardon me for this error. And so okay, I didn't have say. So the and option D, the D-cycloserine inhibits the alanine racemase. Okay. So you have to identify the correct statement among these four. So the correct statement among the following is the D-cycloserine inhibits the alanine racemase. Okay. Now let's see what is all about. So this is the structure of the D-cycloserine. Okay. And this D-cycloserine, it is actually there is a it inhibits the alanine racemase, and this alanine racemase, you can see there is a the reaction cycle here, and this inhibits this there is a oxim is released here, and there is the isoxazole bound enzyme first, and the PLP, it helps to the reactivation of the alanine racemase. after the inhibition. Okay, the dicycloserine, it inhibits the alanine racemase, and the PLP, it reactivates the alanine racemase, and this is the structure of this alanine racemase. Okay, and actually this alanine racemase, it is used in the first line of treatment of tuberculosis. Okay, so please just remember this structure of the dicycloserine. Okay, so this is the structure of dicycloserine, and you know this, for the serine there is a OH in the side chain, and after the cyclization it is called the dicycloserine, and there is a actually I will just show you. Okay, okay, so I think it is clear which is the correct statement regarding the dicycloserine, and the dicycloserine, it is actually does not you helped in the first line of treatment. Okay, now we will just move to our problem number 11, and it says that the problem number 11, it says that how oxalidones work. The options are: number A, it inhibits the protein synthesis by binding to 30S ribosome. Option B, it inhibits the protein synthesis by binding to 50S ribosome. Option C, it unwinds the DNA double helix, and option D, it inhibits the DNA guidase enzyme. Okay, so you have to oxalinidones, they are important class of antibiotics, and they are actually not beta lactamase kind of, they are another class of antibiotics. They are called non beta lactamase kind of, non penicillin kind of antibiotics, and you have to identify what is the mechanism of action of this oxalidones. Okay, so the correct answer is option B, it inhibits the protein synthesis by binding to the 50S ribosome. Okay, so now let us discuss the mechanism of this oxalinidones. So as you can see, this is the all about the oxalinidones. So the examples of this kind of antibiotics are linazoids, radizoid and torozoid. And the linazoid is totally synthetic antibiotic and active against gram positive organism including staphylococcus, streptococci and enterococci, gram positive anaerobicocci, gram positive rods, such as these kinds of antibacteria. And the method of action is it inhibits the bacterial protein synthesis by binding to the 50S ribosomal subunit near the interface with 30S. Thus the formation of the initiation complex is prevented. Okay, so as we all know, in the previous in our previous weeks we have discussed in detail how the protein synthesis by the ribosome and takes place, and in the ribosome we have two subunits, the 50S and 30S, and there is an interface of these two subunits where the initiation complex is formed for the protein synthesis, and the oxalinones, they just bind in the interface, and by binding to the interface in the 50S ribosome, so the formation of the initiation complex which is prevented, and that's how they actually works, that's how they prevents the protein synthesis in case of the bacteria, and they actually kill the bacteria. So as you can see, this is the active site structure. I will just point it out. Okay, so this is called the oxalinidone. Okay, this group, actually the structure of the oxalinidone, it is a heterocyclic molecule. So the structure of oxalinidone, it is like hydantoin. In our previous classes we have discussed about hydantoin, and like hydantoin, this is the oxalinidone structure. Okay, for hydantoin we have NH here, not this oxygen, and oxalinidone, it is a 5-member ring which has this kind of structure, and it is the main group, main moiety for the activity. This is a pharmacophore for this oxalinidone antibiotics, and there is this is the ring A. Okay, in this, so what is the role of the ring A? So the interaction with the peptidyl transferase center, the PTS center of 50S ribosome subunit. So this is the role of this ring in this class of antibiotics, and there are several side chains in this kind of antibiotic. There are 3 side chain. This the purple one, it is the B side chain, the green one, it is the C side chain, and this red one, it is the D side chain. Okay, so what is the role of this all side chains? As you can see, the role of this B side chain, the one is purple, so the role is the interaction with the PTC pi stacking. Okay, so there is a So what is the role of this side chain? This in the red, it is the role. This is not the D ring. Okay, I have just made a so just erase it. So this is the side chain, it is not the D ring, it is the side chain. And what is the role of the side chain? It is very important because they take part in the hydrogen bonding of the phosphate group of the on a residue in upper part of the PTC. Okay. So and what is the role of this ring B? The one is in the purple, the interaction with PTC by the pi stacking. Okay, what is the role of the ring C? The ring C, it is in the green, as you can see. So what is the role of this ring C? The ring C, the role is modulation of the pharmacokinetics. Okay, and what is the role of ring D? Ring D, as you can see, there can be another ring present with this ring C, or there can be a side chain. So it can be referred to the ring D or the side chain. So what is the role of this side chain or ring D? It is to improve the lipophilicity. And what is the lipophilicity? And previous lecture we discussed the lipophilicity, which is related to the partition function, which is given by the log P value. And by the Lipinski rule, we know that there is a certain value of the log P which is needed for a molecule to be a drug, and this value is the measure of the lipophilicity, and this lipophilicity is maintained by this side chain D or this part of the molecule. Okay, so it is all about the oxydones, and this is very important to remember what is the role of each and every side chains. Okay, only remembering few examples and the method of action won't be enough for this molecule. You need to remember what is the side chains, what is the rings involved here. There is a important ring A, which is the oxalinidone ring itself, which is involved in the interaction with the PTC center of the 50S ribosome unit, and with it we have this side chain, this CH2NH-COCH3 side chain, which actually takes part in the hydrogen bond with the phosphate group on the residue upper part of PTC. The ring B, which is involved in pi stacking interaction. Ring C, which helps the modulation of the pharmacokinetics, and ring D or the another side chains, which are helped in the improvement of the lipophilicity. So as you can see from these examples and the structures of this oxidonidones examples, as you can see that this part, this side chain in the red, in this oxidonidone in blue, and this fluorobenzene kind of ring in the purple. So it is common in every examples of the oxidonidones. Only based on this ring C and ring D, there examples have created, and we have several different side chains here, and the compounds are classified because of that. Like if we have this kind of triazine kind of bond molecule here, we will call it delpazolide. And if we have a pyridine molecule with a tetrazole, it's called the tedizolide. So these are the different examples. You don't need to remember exact structure for each and every example of oxidinidone, but please remember the basic structure. Means the ring A, the side chain, ring B, and there is a ring C. So the positions and the basic structure you need to remember with their actions. Okay. So I think it is clear what is the oxalinidones and how they work, and what is the role of each and every subunits of this molecule. Okay, and the method of action. So now I will move toward our problem number 12. Okay, so the next problem, it says that find out the incorrect statement among the following. Option A, moxifloxacin is a quinolone class of antibiotic drug. Option B, method of action of quinolones is the inhibition of DNA guidase. Option C, doxycycline works by stopping DNA replication. Option D, tetracyclines prevent the binding of tRNA with the A side. So you have to identify the incorrect statement among the following, and there are several class of antibiotics are involved in this question. There are mainly two actually, which are quinolone class and tetracycline class of antibiotics, and regarding these two class we have these four statements, and you have to identify the incorrect statement among the following. So let's first see the incorrect statement, then we will discuss about this two class in detail. So the correct statement is which is the incorrect statement, the correct option I should say. The correct option is the doxycycline works by stopping DNA replication. So it is the incorrect statement among the following. Okay. So what is the mechanism of action of this moxifloxacin? Okay. Okay. So as you can see, this is the doxycycline molecule. Okay. and this doxycycline molecule, they actually works, there are belongs to the tetracycline molecules. So what is the role of this tetracycline molecules? Actually tetracyclines prevent the binding of tRNA with the A site. Okay, so this is the, actually you can see there is a 50S this is the ribosome, bacterial ribosome, as we were discussing in our auxolinidones also. There is a 50S subunit and a 30S subunit. Okay, and there is a mRNA triplets template where it binds the place, and there is a there are two sites among this 50S subunit. There is a P site and a site. Okay, and from, as you can see in the, from this P site, the we will just on the pen. So as you can see from this P site, there is a nascent polypeptide chain is formed, but when there is a tetracycline molecule, it just attached with the A site, and when it is attached with the A site, the protein synthesis is inhibited. Okay, so the tetracyclines, like oxolinidones, they also hinder the protein synthesis in the bacteria by binding to the different sites, like in case of the tetracycline, they bind to the they prevent binding of the tRNA with the A sites, because tetracycline itself it bind to the A sites. And there are few examples of the tetracycline molecule. Doxycycline is one of those. And there is another class of antibiotics which has been discussed in this problem, which is the quinolone class of antibiotic. So let's discuss about quinolone class 2, because you can have the problems related to the quinolone class in your finals. It is very important class of antibiotic, and the mechanism of action of quinolones is the inhibition of DNA guidase. So okay, so DNA guidase is a very important enzyme, and the mechanism of action of the quinolones is they block the bacterial DNA synthesis by inhibiting bacterial topoisomerase 2, which is the DNA guidase enzyme, and in topoisomerase 4. So inhibition of DNA guidase prevents the relaxation of positively supercoiled DNA that is required for normal transcription and replication. Inhibition of topoisomerase 4 probably interferes with separation of replicated chromosomal DNA into the respective daughter cells during the cell division. So this is the mechanism of action of the quinolones, and you can see there is a, this is the structure of this quinolone molecule. Basically quinolone, there is a, this is the we know the structure of quinoline. Okay, so there is a structure of quinoline. So all this this antibiotics, the names comes from the structure. As in case of oxolinidones, we say that the active site we have oxolinidone core, from which we have the name of that group. In the tetracyclines molecule, like doxycycline, we have a tetracyclic, means there is a 4 cyclic group here present. As you can see, there is a 4 cyclic groups are present in the doxycycline, so they are called tetracyclines because of that. And this is the structure of the quinoline, and this is the structure of quinolone. Quinolone means we will have a ketone group here. So this is the site, this is the quinolone group, and by depending on this quinolone pharmacophore, this antibiotic class got its name as quinolone. And these are the few examples of the quinolone. There is a ciprofloxacin, moxifloxacin, there is a ciprofloxacins, levofloxacins, and there are basically this quinolone group. We have this quinolone in each and every molecule, but the side chains are different, and based on the side chains, different side chains, we have different names for the drug molecules. Okay, so I think it is clear. Please, this is very important to class of antibiotics, this quinolones and tetracyclines. Please remember their mechanism of action, how they work, and few examples, and it will suffice. And also please, I will suggest, remember the structures of this tetracycline, one or two tetracycline or quinolones, because you can be given just only the structure, and you need you can ask to you can be asked to write the name of the antibacterial class from which this molecule belongs to. Okay, so if there are 4 cycles like this, it will call the it will belong to the tetracycline class, and if you have this kind of quinolone core, then it will belong to the quinolone class of antibiotic. And if you have this oxolinidone group core, then it will belong to the oxolinidone antibiotics. Okay, so I think it is clear, this problem, and I will now move toward our next problem. So the problem number 13, it says that erythromycin belongs to dash class of antibiotic. You need to identify the class of antibiotics in which the erythromycin belongs to. The option A, macrolide. The option B, tetracycline. There is a typographical error here, I will just correct it later. The option C is oxalinidone and option D is quinolone. Okay, so you have to identify the class of antibiotics in which the erythromycin belongs to. So let's see the class, and the class of antibiotic is the macrolide. So erythromycin, it belongs to the macrolide class of antibiotic. So this is the structure of the erythromycin. So like oxalinidone, tetracycline and quinolone, erythromycin is also a very important class of antibiotics, and this is the structure of the erythromycin, and it is called the macrolide. And why it is called the macrolide? Because we have a very big cyclic core here. As you can say, there is 1 2 3 4 5 6 7 8 9 10 11 12 13 carbon cyclic structure we have, which have a 13 carbon, and this is you don't need to remember this structure of this macrolide at all. Just please remember, if you are given this kind of structure, it will belong to the macrocyclic kind of antibiotics. So let's see what is the mechanism of action of the macrolides. So erythromycin, it displays a bactericidal activity, particularly at higher concentration. In previous slides we discussed what is bactericidal. It basically kills the bacteria. Okay. It prevents the growth of the bacteria by inhibiting their protein synthesis. Erythromycin binds to the 23S rRNA molecule in the 50S subunit. The binding blocks the exit then exit the growing peptide, thus inhibiting the translocation of the peptides. Okay. So in this figure you can see there is a 50S ribosomal subunit where we have the place of the translocation, where the erythromycin, which inhibits this translocation and of the peptides, and thus they inhibit the protein biosynthesis in the bacteria. So if you are asked which of the class of the antibiotics which inhibit the protein biosynthesis in the bacteria, you should remember there is the macrolide class, and there is oxalinidones, and also there is a tetracycline class. So these three class of antibiotics, they inhibit the protein biosynthesis in the bacteria, and thus their mechanism of action is somewhat related, because both although they are different, because each of them bind to the different sites and inhibits the bacteria, and only the quinolones, they inhibit the DNA biosynthesis. Okay. So I think I am clear about this. We will quickly move toward our next problem. So the problem number 14, it says that which metal is present in the active site of isopenicillin N synthase? Option A, magnesium, option B, iron, option C, cobalt, and option D, nickel. So let's see. The correct answer is option B, iron. So iron is present in the active site of isopenicillin N synthase enzyme. So this is the active site. As you can see, there is the isopenicillin N synthase is also abbreviated as IPNS. Okay. So there is a first there is a ferrous active site in absence of the substrate. And what is the substrate? The substrate is the ACV. And what is the ACV? In the previous lecture we discussed the active the three simple subunits of the penicillin, which were the aminoadipic acid, the cysteine and valine. So they are abbreviated, their combination is abbreviated as ACV, which is the first substrate, and it is the substrate for this this ACV binds to the iron site with this sulfur of the cysteine and sulfur, for this cysteine and sulfur which is present there. And you can see that in the active site there is iron, which with which there is a histidine, two histidine molecules, one aspartate residue, and one asparagine residue is present here. And when there is the substrate is formed, this it will be pentacoordinated, and at first it was a hexacoordinated iron, ferrous ironous. Okay. So there iron, the what is the you can also be asked about the charge on the iron. So since this is the ferrous, the charge will be 2 here. Okay. So you don't need to remember the structure of this isopenicillin N synthase at all. Just remember that in the active site there is a iron present here. Okay. So I think I am clear about this question, and now I will move toward our last question of today's session. So the problem number 15 it says that IPN to 6-APA conversion is done by option A ACVS, option B IPNS, option C DAOCS, and option D IPNA. So these are the all abbreviation of the enzymes. Okay, I will suggest you the correct answer is IPNA. I have just shown you the correct answer first, then so that we can discuss in detail. Okay, so it is the catalytic cycle, you can see. So there is a L alpha amino adipic acid, L cysteine and L valine present. So the first step is the formation of ACV, and the formation of this ACV, it is catalyzed by ACVS, which is stand for ACV synthetase enzyme. Okay, as I said, please I will suggest please remember the abbreviations also, because not all the time you will given the full name of the enzymes. You can be given abbreviations like this, and you need to understand that it is very easy actually. So ACVS, you know, ACV is the stands for this L alpha amino adipoyl L cysteine D valine, and this synthesis is catalyzed by this ACVS synthetase enzyme. So next we will have this uh, IPNS. So what is the role of this IPNS? So basically the role of this IPNS is the isopenicillin N synthesis. The role is synthesis of isopenicillin N, which is the IPN. So this is the structure of the IPN. So from the ACV to IPN conversion, it is catalyzed by this IPNS. So next step we have this IPNA, which is the IPN amino hydrolase or acyl transferase enzyme, which catalyzes the conversion of IPN to 6-APA. The 6-APA stands for 6-amino-penicillinic acid, which is a very important molecule. Okay, and this IPNA then again can work on this 6-amino-APA to form the other penicillins, and from this IPN, after the use of the IPNE, which is the IPN epimerase enzyme, which converts the IPN to the penicillin N. Okay, and DOCS, which stand for the DOC synthetase or DSC synthetase. So they work on the formation of this DAOC, which is the deacetaloxycephalosporin from the penicillin N. Okay, so this is the very important catalytic cycle, and I will suggest you please remember this catalytic cycle. It is very important how the penicillin N is formed, how the cephalosporin is formed, and which is the substrate. So which is there is a ACV, there is a IPN, 6-APA. So these all are very important. You don't need to remember the each and every structures. If you can remember the structures, which is very good, because the structures are very important, and there can be questions regarding from the structures. Okay, you can be given this structure and can be asked which is the what is the enzymes involved in this conversion. Okay, so this question can be given you like this also, without giving the names. You can be only given the structures, and you have to identify the correct answer. So then if you can remember, that's why I am saying, if you can remember the structures, which is it will be very beneficial to you. And please remember the names and the abbreviations 100%, they are very important. I think it is clear that the ACV formation is catalyzed by this ACVS from this three components, this alpha amino adipic acid, L cysteine and L valine. This IPN formation from ACV is catalyzed by IPNS, which is IPN synthetase, and 6-APA formation from this IPN is catalyzed by IPNA or IPN amino hydrolase, and the penicillin N formation from IPN is catalyzed by IPN epimerase, and this cephalosporin formation from penicillin N is catalyzed by DOCS enzyme. Okay, so it was all about today's session, and I think okay, we have reached to the end of the session. And if you have any query, any problem regarding any question, you can email me. So I will just share my email ID with you here. Okay. So this is my email ID. So regarding today's session, if you have any doubt regarding any problem, any question, please write a mail to me. I hope I was clear about the session of what we learned today. and this slides, this PPT and this lecture video will be shared to you by the in the NPTEL website. You can download it from there, the slides, and you can read it better. So this was all about the week 10 session, problem solving session of the NPTEL course, and I thank you for joining today's class. And then next Friday we will have same meeting from the 7 p.m. to 8 p.m. and then next Friday we will solve the week 11 course contents related problems. Okay, so now I will close the session for today, and thank you for joining it, and let's meet in the next week. Okay, thank you so much. Thank you so much all. Thanks all. I will close today's session.